Well,
Hi, how are you?
would you know anything about how to use Word and
Uh,
numbering?
Numbering?
Yes.
A little bit. I mean, you have to go to the menu and number, and you can like configure the numbers
Yeah.
in the
Well,
footer.
I'm doing a thesis, and there's something called the front matter, and it's in Roman numerals, and then you have to switch over to the other to regular Arabic numerals for the rest of it. How do you do that? Because
So
I
yeah, you have to do a page break or a document break or something. I think they call it a page break. So, or no, I guess
I did.
yeah. So you do the first part, you break, and then you do the second part.
Yeah, I know, but um, it didn't work.
Well, okay. So I don't have it in front of me, but there's it's not actually because there's a page break which is where you break to a new page, and then there's like I guess like a
Sorry,
document.
I can't hear you. This thing has gone off beside me. Say that.
All right. So yeah, they have like a page break, which is where the
Sorry.
page.
Sorry. Okay, somebody answered it. Can
Okay.
you please tell? Okay, what did you say?
So they have like a page break, which is the where you can just break to the next page, and then there's a document break where you actually break the document like into two documents, and you can like start off like on the next page, but it starts the document over, and it's
So
different.
maybe I need to look for document break.
I think so. Yeah, you'd have to look
Uh,
in. Yeah, you'd have to look up in the doc, like in the. Let me look. I can check here.
Because a very snippy professor saying there's no time for coaching, and we're going. The only problem I have is that I can't get the page break to work
Right.
to start the numbering
So it's
over.
actually called section break.
Section break. Okay. All right. Maybe that's it.
I think so.
One fellow wants me for section break. One fellow wants me to use a passive voice, and one wants me to put I in the abstract.
Yeah.
What's the convention?
Well, the way I usually do it is the active. So like it's like I guess you could say I because you're not working with colleagues. You could put we if you had like co-authors. But it's usually present tense. So like so I conduct or maybe conduct an analysis instead of an analysis was conducted. So you do the present tense. You can use I because usually in papers people use we, but that's with co-authors. So
So
it's usually
that the one professor likes the two I's I put in it in the abstract, and the other fellow says, "No, I want a passive voice."
yeah. Yeah.
I just like don't know.
Well,
I
that
don't
seems like
know.
more of a problem of they can't agree. You have to get them to flip a coin.
Oh well, I'm not sure what to do about that. But anyway, I'll try for a section break.
Okay, yeah, that sounds good. That sounds good.
Thanks. I just like this. It's probably all like wrong anyways, so I'm going to have to fix it. But
Oh,
like for numbering my equations, I just put 1, 2, 3, 4.
yeah. Well, that's how I usually do it. I mean, in the brackets.
Yeah, in the brackets. Anyways, I'll just put the page numbering in and sort of fix the abstract and handed it.
Yeah.
Yeah.
Yeah, it's always that's Well, one of the big challenges of like thesis writing is like getting Word to do things that you want it to do, like for formatting.
Yeah. Well, my guy said, I can't coach you. You hand it in today. It's like, really? I've been working on the stupid page break thing for all weekend.
Yeah, yeah,
Doesn't matter which way I twist my tongue, it still doesn't work. So it must be a section break.
yeah.
Anyway, never mind.
That's okay.
It's just foibles.
Yeah. Okay. Well, welcome. So it looks like Morgan's here. Susan's here. Um, yeah. So any questions or issues you'd like to raise before we begin?
I wish I knew where to find more data or, well, graphs of tissue when they're being tensioned or compressed. I really would like to. I've got about 4 or 5 examples, and it was hard to find. And the one thesis I looked at was they were doing compression, and they literally said, due to the lack of literature on this subject, I am doing this project. Yeah. Maybe I should put that in my conclusion.
Yeah.
Why don't I have? Yeah. I can quote the guy. Anyways.
All right. Yeah. Well, yeah, that's always a thing with new areas is you have to find the right data. Okay. So, yeah, why don't we get started? Yeah, so it's almost the end of the year. I think we'll have one more meeting after this. I think this is a pretty good year. We had a lot of interesting things we talked about. A lot of kind of a lot of themes to stretch out. I've done a little bit of like kind of recounting of what we've been doing this year when I was doing preparing for the annual meeting, OpenWorm annual meeting, and yeah, it's interesting. We had a number of themes. I don't have them with me, but I will maybe prepare something maybe for next week. Um, we started off the year. We were talking about different open source initiatives around OpenWorm and DevoWorm. We did a lot of work with the Google Summer of Code with topological type models and hypergraph models. And then we've been talking about tensegrity and cell mechanics for most of the year. And then not to mention a lot of the stuff with molecular biology and development. So there are a lot of themes. I'll maybe get to that next time, or maybe put out a video on it on YouTube or something, just kind of going through those themes and where the sort of the interests lie, because you kind of go from week to week and you don't know where everything's accumulated. But I'll do that maybe next time. So that's good. Yeah, so Morgan, and we were having a conversation in the Slack about the OpenWorm annual meeting, and specifically a project that is sort of parallel to what OpenWorm is doing. And we'll get to that paper in a minute. But at the OpenWorm annual meeting, we had this discussion, or a lengthy discussion, about some of the other initiatives that people were doing around C. elegans. There's a Chinese group, and I know we covered this paper, that are doing things with some of the OpenWorm tools that is interesting. I think we talked about that around the middle of the year, where they were doing work on, they were kind of trying to develop their own tools, and then they kind of used OpenWorm tools, and then so the people at OpenWorm are aware of that initiative. And then there's this other initiative by a bunch of people, like Adam Marblestone, Conrad Cording, Netta Cohen, who's actually on the OpenWorm Scientific Advisory Board, and a lot of other people on this, they'll call it reverse engineering the worm, which we'll get into that paper and talk a little bit about what they're doing. We talked about this as well a while back. It was kind of an informal discussion that I think Conrad Cording brought up. And he put something out like a Word document, and it was kind of rough and ready. So it wasn't like anything formal. This is a more formal uh, statement about it. What they wanted. It's a vision paper where they talk about how to not only build a model of C. elegans, but also to do experiments in terms of the connectome to look at how each neuron functions in the context of different manipulations. So you'd have a lot of what they're thinking of is really kind of bringing optogenetics to bear in stimulating individual neurons and looking at different circuits and what behaviors they generate. So it's very interesting. So this is the paper here. The time is ripe to reverse engineer an entire nervous system, simulating behavior from neural interactions. This is a number of people. Gal Haspel is the first author. Ed Boyden is on this. George Church, Netta Cohen, as I mentioned before. Daniel Colon Ramos is at Yale. He's a C. elegans person. We have, let's see, Eduardo Izquierdo, who's worked with OpenWorm in the past. Sean Lockery, who's a C. elegans person. Adam Marblestone, who does a lot of synthetic biology. Uh, let's see, Ellie Schleiserman, who's a C. elegans person. Avatar Yamini, who's worked with OpenWorm in the past. And Conrad Cording, of course, does a lot of the stuff with Neuromatch and that. And then Manuel Zimmer, who's also worked on with Avatar Yamini on some of these things in the worm. So there are a lot of C. elegans people, synthetic biology people, and neuroscience people. So this is really interesting. They have, we talked about another vision paper where people talked about using like deep learning for something or another, and it was like, the vision paper was basically stating your vision for something. It didn't really have a lot of like, we're going to actually show how this is done. It's just kind of like, this is what's needed. So just to give that context. Okay, so the abstract reads, so just like electrical engineers understand how microprocessors execute programs in terms of how transistor currents are affected by their inputs, neuroscientists want to understand behavior production in terms of how neuronal outputs are affected by their inputs and internal states. So, we think about, so this is interesting context. Conrad Cording was a co author on a paper several years ago um, called Could Neuroscientists Understand a Microprocessor? And the idea was if you took a neuroscientist with training in looking at circuits, neural circuits, and you drop them in front of a microprocessor and they had never seen a microprocessor before, could they figure out how the microprocessor works by kind of studying the output, studying what the microprocessor does internally, and then providing inputs to the microprocessor. So that's how this is kind of the idea here. So they're kind of building on that idea, but basically, if you think about what neuroscientists do when you do experiments, behavioral experiments, is you might have like two different strains of a mutant, or you might have a wild type and a mutant. You might have some condition where you have like an expert and a novice, or you might have you know like some sort of neurodegenerative disease and a normal phenotype. So you have these differences that you use in an experiment. And it's really about experimental design. So you have these different conditions, then you have some common behavioral task, you keep that controlled, then you have the behavioral task that's performed, there's something that goes on inside, and then there's a behavioral output. And you compare the behavioral output, and then you try to infer what's inside the brain, trying to, that make accounts for this difference or a non-difference. So, we can use different neuroimaging techniques or whatever to get at that. And, we can also use optogenetics to stimulate certain neurons if we wish. But the idea is that we can manipulate things, we can look at the behavioral output, and then we can try to, using those together, to try to get to like an answer of how that circuit works. So it really isn't that much different than how like neuroscientists do science. I don't know why they had to use the electrical engineering metaphor, but maybe it's cleaner. Anyways, this dependency on neuronal outputs on inputs. So neural outputs are dependent on inputs. So if I give you an input that is, you know, of a certain strength, of a certain, amplitude, I should expect some sort of output that may be, proportional. And, there are nonlinearities in the brain, so I don't want to discount that. But basically, they're making this case that inputs condition outputs, or outputs are dependent on inputs. Uh, these dependencies can be described by a state-dependent input-output, or I/O function. However, to reliably identify these I/O functions, so their goal is to create these I/O functions, we need to perturb each input in combinations of inputs while observing all the outputs. Here we argue that such completeness is possible in C. elegans, a complete description that goes all the way from the activity of every neuron to predict behavior. So I think they're kind of arguing for sort of a Turing completeness of behavioral experiments. In other words, in Turing completeness, you have like a set of functions that produce all possible outputs. And usually in like computer science, it's Turing complete. So you can, like, if a simulation is Turing complete, you can simulate the entire system or the entire thing that you're trying to simulate. In this case, this is where we have this sort of behavioral Turing completeness, if we want to call it that. It's really maybe behavioral completeness or something like that, where you have enough data on the different perturbations that allow you to observe all outputs. So, you don't have any missing states and you don't have any unexpected outputs. So that's what they're trying to do. And so they're going to build an experimental test bed. We characterized the connectome and C. elegans well enough so that if you kind of have this experimental framework, you should be able in principle to do this. Here we argue that such completeness is possible in C. elegans, a complete description that goes all the way from the activity of every neuron to predict behavior. So we have the information about every neuron in the connectome. We have a lot of information about the activity. And what we want is this complete description of all the activity and in a way that predicts behavior. The established and growing toolkit of optophysiology, and by that they mean optogenetics and other optical stimulation techniques, other optical recording techniques, can non-invasively capture and control every neuron's activity and scale to countless experiments. The information from many such experiments can be pooled while capturing the inter-individual variability. So they're interested in taking sort of the the ability to sort of control every neuron's activity, to turn it on and turn it off at will, to record all the different sort of contexts that are needed to predict behaviors, and then also to look at inter-individual variability. So that, we have differences in a lot of C. elegans. We have mutants, defined mutants. We have wild type strains. We have males and hermaphrodites. We have different, just kind of inter-individual quirks of C. elegans. We talk about C. elegans being sort of this, deterministic, having this deterministic biology, but there are differences sometimes in how the brain produces behavior. So there are a lot of things we can, a lot of sort of individual variability things we need to account for. Okay, so because neural identity and function are largely conserved across individuals, but not, it's not complete conservation. What they mean by conservation is that neural identity and function don't change across individuals. And, unless you're dealing with a mutant, or sometimes in males, it's different than hermaphrodites. So just like electrical engineers use transistor I/O functions to simulate program execution, we argue that neuronal I/O functions can be used to simulate the impressive breadth of brain states and behaviors of C. elegans. So their argument is basically that it's not really building a simulation per se, it's building the conditions for a simulation. So they're backing up from what OpenWorm is kind of doing and saying we can actually provide this framework for, we have an organism that has some individual variation, but not too much. We know the connectome, we know kind of what it does. So we can apply these technologies to sort of manipulate the internal state enough so that we can produce all sorts of behaviors. But more than that, we can produce these input-output functions so that if I give you some input, I can predict the output pretty well. So that's what they're trying to do here. So they talk about in the introduction, they actually talk about this paper. Can a neuroscientist understand a microprocessor? And they talk about for such a simulation, one needs a list of all components or a net list and the input-output functions of all the elements. The simulation is then the key tool for understanding information processing in the microprocessor and asserting that it is correctly engineered. So the idea is that you have to sort of provide a list of components and input-output functions to a simulation. And from that, you can then you can understand what a microprocessor does. You can understand all the output states and where they come from. Then they have this part. When philosophers talk about understanding and explaining, they usually distinguish many kinds thereof. According to Tinbergen, there are 4 categories of explanation. So these are Tinbergen's 4 explanations, divided along axes of proximate to ultimate and static to dynamic. So we want to maybe say understand the phylogeny of the nervous system, the evolutionary trajectory that made the nervous system the way it is. Another goal is to understand how development or ontogeny gave rise to the individual animal as it is now. So within the lifespan, how did that nervous system develop? A third goal is to understand the function or adaptation of the nervous system. So, once the nervous system is, say, an adult form, how does it adapt to physiological conditions, to environmental conditions? And then lastly, the goal is to reveal mechanism or causation. So how does something actually work? What comes before what? What causes what? This goal is usually taken as describing the function of something in terms of causal interactions between the involved components. So that's exactly what they're doing. And then, of course, the model of the understanding the neural processor is very much like how other sciences find relationships between different things. So they're drawing inspiration not only from electrical engineering, but from chemistry, from physics, and so forth. So when neuroscientists aim to understand brains, following Tinbergen's 4 questions, they thus use many different levels and definitions of understand. For some, understanding means the ability to fix diseases. For others, understanding means knowledge of involved representations. And yet for some, understanding means the ability to simulate. An analogy to the electrical engineering case, the ability of faithful simulation would then also come with solutions for fixing problems, predicting the nature of brain representations, and a range of approaches to deepen our understanding of the brain. So that's where they kind of leave it for that. And then, they kind of talk about why you should reverse engineer a nervous system. So Um, so a central goal of systems in computational neuroscience is to model how nervous systems convert stimuli, spontaneous activity, and internal states into things like coordinated muscle contractions and other types of behaviors. So their inputs to behaviors are kind of like raw materials. And then there's this product, which are, different coordinated behaviors. So you might have, say, light stimuli, you might have some spontaneous activity among the neurons, and then you might have internal states which are not spontaneous, not stochastic, they're actually like intentional states. And, a light stimulus might say condition this internal state into some sort of response paradigms or response mechanism. So you might have an internal state and you might have spontaneous activity, and the presence or absence of light will turn on or turn off this internal state. So you end up with two states on and off. You end up with spontaneous activity that might actually also tip the system from one state to another, or it might interfere with the stimulus. But in any case, you have these three components. And then those things lead to things like coordinating muscle contractions or other types of behaviors. So, that's kind of the idea. They actually talk here about the Brain Initiative, where, we're trying to develop new large scale neurotechnologies, and other funders like the Human Brain Project, Microns, and the Simon's Global Brain Collaborations, where they're also trying to reverse engineer the brain. So and then success in reverse neuroengineering is actually having this kind of, ability to predict outputs from inputs. So, um, let's see. We consider each neuron a muscle cell to be computing its output as a function of the activity of the input cells and spontaneous activity. So it's, you have output equals input plus some stochastic term, or it could be multiplicative if you wish. But basically, that's the equation that we're using. Um, and then, as such, it is characterized by a spatiotemporal function mapping input traces into an output trace. So you have the spatiotemporal uh, input, which is space plus time. And then you have this output, which is this output trace, which is, I guess, also spatiotemporal in space and time. So if you have an input of light stimulus, the light stimulus has a source in space. It can turn on and off with respect to time. And then that converts through this uh, stochastic function, this random function, which adds noise to the system. And then you end up with this output that also is located in space and time. So if there's a source of light, to my, 30 degrees to my left and, up above the horizon, I'm going to move towards that light source, perhaps. I coordinate my limbs, I move towards the light source. And sometimes there's noise. Like, for example, sometimes it's confusing as to where the light source is. There might be multiple light sources. So you have to decide which light source you want to go to. And that's how you get this sort of input output. The input is spatiotemporal. It gets processed in terms of this function, in terms of noise, and then you have the spatiotemporal output. So that's what you're trying to reverse engineer here. And then, of course, you have to think about the output trace IO function, which represents both synaptic and non-synaptic effects. So this mapping is very simple in terms of an equation, but then the equation, you can unpack this into, you can unpack this stochastic term into like a stochastic term and then some synaptic and non-synaptic things. So you have, say, for example, if you had like a neural network, you would have like some stochastic term plus maybe the weights of that network, which would be very analogous to synaptic function, and then non-synaptic effects, which would be something else that we could use to inform the network, which we usually don't use in neural networks, but we could put one in. So the non-synaptic effects are things like neuropeptidergic, signaling, or spontaneous activity, because we could use the random noise, we could use some other set of terms to fit that together and then say that plus the input gives us the output. So again, I don't think it has to be additive because a lot of this, this is all processing, and so it's probably there's some nonlinearity here. In this reductionist framing, reverse engineering consists of figuring out the input-output mapping for all neurons and muscle cells, as well as the inputs from the world, or system identification, thus reassembling the collection of I/O functions into a robust, simulatable model that it can exhibit key behaviors when connected to a simulated body. So in figure 1, they show this. Moreover, behavioral variability will be recapitulated by drawing parameters for multiple simulations from the empirical variability of I/O functions. To be deemed successful, this working model should recapitulate behavior and behavioral variability under a range of conditions, stimuli, and perturbations. So figure 1 is down here. Right here. So this is their model overview of the proposed approach, demonstrating how imaging, recording, and perturbing modeling and simulation interplay. So the first thing they want to do is do imaging. They want to image a bunch of worms. You can image the nervous system. You can do this optogenetic stimulation. Well, the imaging is really just to get a co-register a lot of the things they're going to be doing with recording and perturbation. But basically, there's an imaging component. Then there's recording and perturbation. You can do that during the imaging step, where you're recording from each neuron, you're perturbing it with light, you're getting the sense of how the neuron responds. Then you have this modeling and evaluation, and they build their functions to predict the model outputs, but then they also have this ground truth, which is the recording and perturbation step. So you get a bunch of data from the imaging. You get model outputs. You put this into a model. You get the outputs. You simulate those. You also have a ground truth, which you observe from the imaging, and then you try to compare model and experiment. And then, of course, simulation. You further put this into like a digital model of a worm, and then you do this real world behavioral validation where you compare it to the real worm. And you likewise do this model versus experiment stuff. So that's where we have figure 1. So they propose a bunch of things that we need to do for this. Again, this is kind of very C. elegans like. This is a lot of stuff that you know OpenWorm has been doing for a while. I think that the innovation here is kind of the manipulation of the behaviors, getting different behavior, getting this catalog of behaviors basically, and making this more or less Turing complete. That's what they're proposing. As far as I can tell, anyways. I may not be doing it justice, but I think. And then they have this bit about power calculations. So this is where you're really trying to get statistical power, and you're trying to figure out how much experimentation you need to do, not just to produce these functions, but to get some statistical power so you can make statements about these things instead of just kind of showing a proof of concept. And this is where it gets interesting because, with a lot of behaviors, to really characterize them fully, you don't just need a bunch of different behaviors or all of the different behaviors that exist, but you need a lot of observations of them. So you know that something is not just a statistical anomaly or, it's, just kind of some random behavior. You can determine random behaviors from non-random behaviors. So that's where you need your power calculations. You need to figure out how many experiments you need to do, how long those experiments need to be, and how many C. elegans need to be involved in these experiments. So they're really focused on power calculations here. This is where we kind of, if you're familiar with statistical power, you know that this is just a tool to tell you how robust your result will be. once you make a statistical test. So if you make a statistical test and you get a significance and the power of the experiment is low, if it's underpowered, then that significance doesn't mean as much as if your experiment were properly powered and you get a significant result. Basically, that's the idea. Then, they think beyond C. elegans. So ultimately, the value of the reverse engineering approach that they propose here will allow us to do this for other systems. So we might be able to scale to other systems like, fruit flies, Drosophila, or, maybe something like mouse, which, is probably well off into the future. And so this is said just like the sequencing of the first genome enabled by the Human Genome Project and the sequencing of the genomes of many humans, reverse engineering a nervous system promises to open the floodgates to reverse engineering many other nervous systems. So they're using the Human Genome Project as sort of a comparator here. And so, needless to say, we need to think about generalizing to larger nervous systems. A lot of people in this group are interested in human brains, which are much, much larger than C. elegans, produce a much wider range of behaviors, which is interesting because, it's not, and C. elegans neurons don't really have the same type of spiking behavior that mammalian neurons do. So these are all these challenges in kind of scaling this up to humans, or even to something like mouse, or even really to something like Drosophila. But that's where they kind of want to go. So that's basically the idea. It's a vision paper, and those are the references. So that's where they are, and I think it's fascinating. It's a fascinating kind of take on this. I think the main thing they're trying to do beyond what like's been done with OpenWorm is this idea of collecting data for all possible different behaviors. And you can do that in C. elegans, I suppose, if you had enough resources. We have enough community tools to say kind of what the basic behaviors are, what the basic neural response is, but then to kind of work all that out into this complete description is really where the power is in this. All right, so a few words about these input-output functions that they were talking about in the first paper we talked about in the meeting. So, we talked about this term, and I was kind of doing it in my notes, so I didn't show what I was diagramming. But basically, we have we're trying to solve for an output, and in doing so, we have inputs, and we have maybe an additive noise term. So, additive noise term would be like what's going on in the brain, and maybe like a brain network where you have an input here, which is this term, and then an additive noise term, which maybe modifies the input in different ways, provides maybe, ways to dampen the input or accentuate the input or interfere with the input. So we have this noise term plus the input equals the output. And then they talk about maybe a more sophisticated model where you have multiple compartments for the second term. So, of course, we're solving for the output. We still have the input term, and that could be something like maybe like a light source. And this, of course, we said is spatiotemporal. And remember, our output is also spatiotemporal. So we have the output, which is a behavior, which is also spatiotemporal. And then we have maybe an additive term here. But instead of just chalking it up to noise, we can break this down into a couple of different components. So we still have some noise because the neural activity often involves a stochastic component, a noise component. We might also add in maybe something like synaptic, like maybe a weight matrix. So in a neural network, we'd have a weight matrix. In the brain, we might have synaptic activity, and we could summarize that as a weight matrix. And then maybe things like neuropeptides, or like a matrix having to do with neuropeptides. We have neuropeptidergic connectomes in C. elegans that have been drafted. So those are all things that we can put into this term. And these all kind of maybe are additive. along with the additive component of the input, and it gives us the output. These can also be multiplicative, as I mentioned. So all these things can be multiplicative, and it has an effect. It actually is more of a nonlinear effect on the system. So this is assuming some sort of linearity or nonlinearity to what happens inside the brain. So the input is, of course, from the environment, and this other term is internal. So we're really trying to predict the internal state, which is this component here, this compartment of our model. And the environmental component is summarized as input, but we can just as easily break that down as well into multiple components. So we might take our input and break it down into something like a light source, maybe like an odorant, maybe like if we're talking about spatiotemporal variation, maybe it's like a landscape. Like there are certain places where you can go, certain places where you can't, in terms of navigation, in terms of movement. And so those are all things that are in the input. We can actually have a bigger component there. So yeah, I hope Morgan found the discussion as paper enlightening. I mean, we didn't get to discuss it Saturday. Actually, Saturday we discussed some things about kind of like simulating the brain. So Morgan, you had something to say?
Oh no, I was just going to say that what you said was kind of Conrad's pitch,
Yeah.
and he seems to be trying to raise money to collect this data, right? So he was at this meeting because it's definitely a place. Well, he's a Foresight fellow,
Right.
or that they gotten some money to start this on the basis of a Foresight grant, and it's funny because somebody who I was going to meet at that meeting had already shared he'd shared a Conrad paper, a Conrad last author paper from like 2013 that was like scalable neural recordings. Forget that the title. It's in Frontiers. But I said to him, like my take on that paper was like this was like a white paper for the Brain Initiative, this 2013 paper, and I think that was about the right time for it, but I was also, I was looking at this 2013 paper and I was just like, look at these people, like it's Conrad Cording, who, again, I think of as kind of as a neuroimager, whole brain fMRI neuroimager, and but he's on this paper with like all these invasive recording people.
Yeah.
and again, it just struck me as a Brain Initiative kind of paper, but it was useful. I mean, again, like it was only lightning talks at this Vision Weekend, so it was a very short. I think I've got a picture of each of his slides because he only had like 8 slides, but I felt that the video that he had from last year, excuse me, it was like more like 50 minutes and laying out, kind of described, gave a lot more of kind of like his motivation and sort of like how did someone how did a nice neuroimager like him end up proposing to do C. elegans work.
Yeah.
And the so again, it was kind of a combination of him saying somebody needs to do this, right? So one of it was just like, I've tried an NSF grant, center grant, and we got rejected. And and again, like, I look at this paper and I see a lot of big names on this paper, right? And certainly a lot of like you said, like, he seems to have, again, pulled together a number of really high profile people doing elegans work, and with kind of Fermi like calculations, has put together what wants to say is like, this is the data that we need to get to kind of forward solve the neural behavior, and was kind of now pitching a more like, I think he'd said, like, the center grant was going to be like 60 million because he was going to lose half to the institution. And, he's selling it to Foresight people as like a $20 million grant, as maybe like a focused research organization, right? Where it's like that's the entire entity. And so that was cool. And I think he also threw in, just in our pinning him down after, that he felt that it was time for him to do something new.
Yeah.
So he was also like of the opinion that if things start getting stale, like, change. I mean, I wouldn't exactly call this changing field, but he was just like, no, I'm doing something new now. Or, well, he kept on saying, like, I'm trying to switch. I'm trying to pull together, and but it was nice. It was certainly nice. I'd never met him at a neuroimaging conference, and so it was certainly nice to talk to him and see how he phrased it all. And then I don't know if I'd seen this before. I mean, I feel like I do follow Foresight. Like, I feel like I should have seen this, but anyway, but it was nice. And nobody else, the other, I mean, E11 Bio was one of the other speakers, which is a focused research organization, and it's kind of doing cell atlas connectomics, but very much like, somebody came up to him while we were talking and was just like, hey, why aren't you looking at zebrafish or something? And he said, that basically it would take too much data, like this wasn't, and this was, where he'd want to start. And I did ask a little bit about, how sophisticated was the microscopy? Like, what kind of equipment, kind of buy-in would you need to be able to collect the kind of data that he wants to do? And it still seems pretty high end. But it's also the kind of thing where I'm kind of surprised. I mean, like why isn't, or I know Genelia is doing zebrafish,
Yeah, they are.
and I mean, certainly when I remember human in the Brain Initiative being talked about, and certainly like there was this series of talks of kind of like tech people together with neuro people. I forget what that little series was called, but it was a big focus on like scalability. It was like we tech people understand scalability.
Oh yeah.
So can we somehow help? And I remember, but I just remember these, wonderful whole brain zebrafish images,
Yeah.
and thinking, well, yeah. So it was nice. It was nice. Yeah. Like you said, like mammalian is way off, and um, the other people were kind of these focused ultrasound groups, like Mary Lou Jepson, and some are talking about kind of medical devices. So this was the one kind of real ambitious, seemed like the ambitious neuro project. And of course it was et elegans. So I did bring up, I asked him about OpenWorm, and but OpenWorm obviously is not a data generating organization, right? But like you covered there, Tinbergen at least understands that development is one of those kind of explanatory perspectives that you still really need to have.
Right.
So if I could also just add that I got to have a nice conversation with Adam Safran.
Okay. Recently?
And
yeah.
Yeah, at the same meeting.
Okay.
So and that was very cool, because, well, one, it seems like he's maybe, I mean, I think I brought this up that he was pitching at the last one, a kind of empathic AI, and it does seem like he's maybe kind of fundraising for his own AI startup too, which tis the decade apparently. And so he'll be going up to NeurIPS for both networking and potentially fundraising. But what was cool was that he said that he's basically in Michael Evans' lab now.
Oh, okay.
And that they are working on active inference paper that is like active inference in this cellular cellular agency,
right?
And which certainly sounds like maybe some previous Michael Evans papers that I can think of. When I think of active inference as cell, I think maybe Michael Evans may be one of the only people I can think of.
Yeah, there are a couple others, yeah, but
yeah. Well, I would love if you've got any suggestions of others. I'd love to hear them, and certainly, I was gonna, again, this might be Michael Evans covered as well, but I was putting together a couple references that definitely like kind of embryogenesis and how that can be seen as a kind of active inference, certainly of responding to perturbations and things like that.
Yeah.
And there's this great paper that I love of estimating kind of the regulome from spatial transcriptomics data. And it's speaking to kind of, large scale, these kind of large scale genetic networks and their ability to provide some of that cellular inference.
Yeah. I'm just writing some notes here.
Sure.
Yeah. Oh, so yeah, that's interesting. I'm glad that Adam's kind of working on that. That sounds great. Yeah, I think, trying to remember, I think the Active Inference Institute, they were working on a paper about development and active inference. And I can't remember the authors in that paper. I think there are a number of other things that have come out of the Levin lab where they've been talking about, like embryogenesis and not so much active inference, but kind of like mechanisms for, kind of active inference like things. They didn't really use the term active inference. So it's like, it's a framework, of course, that has a formal terminology. So maybe, that's maybe more productive in terms of using the same terminology, mapping things to that terminology. Yeah, I think that's really interesting stuff. And there's a lot of work like, with like genetic networks and gene expression regulatory networks and expression networks and things where you can look at like, how they kind of function and, there are a lot of active inference principles that could be used there. But that's kind of an open area because I don't think people have really looked too much at that specific.
Yeah, that's. I mean, and this that Regulome paper is from Roche. It's like the Roche Institutes in Basel, or Basel, whatever you say. But yeah, that like big like applied organoids research group,
Okay.
which I think is cool. Yeah. I'm gonna definitely have to start shuttling kids in this sec. So,
All right.
but love that you covered that paper, and yeah, would love to follow up on any other references you can think of.
Okay, I'll try to put together something for that. Okay. Well, thanks, Morgan, for that discussion. So I don't really want to get into the other things that I had because it's already almost 10. So for the top of the hour. So I think that's all for this week. Susan, did you have anything to add or?
No, not much. I read somewhere where epithelial cells are not really an organoid, but that you might get epithelial cells on the outside of an organoid if you were going to study them, but that they're usually not studied on organoids. And the measurements I have for some of the one of the papers did cells on flat sheet of gel. So that was interesting. That one is a nice one. I wish there were more like it because they actually showed the cells and then showed what they did. So, yeah, still searching for more data, of course, but pretty difficult.
Yeah.
There's things on the lung at least, and I think these cells were liver cells and back cells. I guess I need to look that up before I hand in the final copy of my paper here. And it's almost ready to go, so I'll hand in version 7 today and then can shred it. I'm presenting on the 16th.
Okay, well, good luck. Yeah, it's coming up.
Yes.
It's a holiday season presentation. How's that gonna work?
Yeah. That's okay.
Yeah.
That works for me.
Okay. Well,
Yeah.
like to
No.
try if you'd like to test it out, next week or whatever in the meeting, they'd be welcome to do that. If
Maybe I could test out my verification stuff since that's what's bothering me.
okay, that sounds good. Yeah.
Okay, I'll see if I get what I do. I'm going to put it together. I'm going to start putting together my presentation.
Yeah. Yeah.
Yeah. So, okay.
All
All
right.
right. Thanks.
Thanks. Talk to everyone next week.
Okay.
Bye.
Bye.
Bye. Okay, now I'd like to go over one additional paper, and this is this paper here, Tissues as Networks of Cells Towards Generative Rules of Complex Organ Development. So we've talked about generative rules before, generative models of development, and everyone's interested in generative AI, but generative modeling has a long history in computing, in generating things from a bunch of interactions, or generating things from, say, like an algorithm, like a genetic algorithm, or even like a deep learning. And it's just basically, defining a bunch of agents or defining a bunch of units and looking at the interactions and what they produce as an output. So they're going to use, and then we talked about how you can apply those kind of generative computational techniques to development. And what development essentially is doing is generating or building a generative model on top of like a gene regulatory network. And that's really giving us this, we have this environmental input, we have the stochastic input, and we have the genome, and it all combines to produce these generative structures, these generative rules. So this is where they talk about generative rules of complex organ development. So these are some authors from a German group, Sabine Fischer, George Bassel, and Philip Holmensberger. So the abstract here reads, network analysis is a well-known and powerful tool in molecular biology. More recently, has been introduced in developmental biology. Tissues can be readily translated into spatial networks, such that cells are represented by nodes and intercellular connections by edges. So we've talked about these kind of networks before, where we can build like spatial networks of cells with intercellular edges. So it's basically that maybe two cells are communicating, maybe they're reinforced by some extracellular matrix, or like we've talked about with our trijunctions and our quad junctions and things like that, where you have these meeting points between the cells that confer some sort of stability. We talked about this in previous meetings, so I won't get into that here. This discretization of cellular organization enables mathematical approaches rooted in network science to be applied towards the understanding of tissue structure and function. In this paper, we describe how such tissue abstractions, so remember these networks are abstractions of a tissue, can enable the principles that underpin tissue formation and function to be uncovered. We provide an introduction into biologically relevant network measures, then present an overview of different areas of developmental biology where these approaches have been applied. We then summarize the general developmental rules underpinning tissue topology generation. Finally, we discuss how generative models can help to link the developmental rules back to the tissue topologies. Our collection of results points to a general mechanism or a set of general mechanisms as to how local developmental rules can give rise to observed topological properties in multicellular systems. So they kind of talk about how network approaches can be used to look at the molecular interactions between cells. A large number of measures enables the analysis of these networks to understand system function. Some of these same methods and analyses can be applied to additional scales of biological complexity, looking at the study of cell interactions in tissue. So they're taking molecular networks as inspiration. Of course, we've done work with cell level networks And that's very, that has a similar sort of structure to it. A perspective of multicellular systems as interacting collectives of cells can be traced back to the work of Ramon Cajal and some of the stuff they've been doing with mammalian nervous systems. And he made a lot of drawings of these wiring diagrams, and they very, very much map to these network approaches. Okay, so this is kind of an example here. This is one of Ramon Cajal's drawings of animal nervous tissue. This is a model of C. elegans in B. In Drosophila, you can cellularize and, a microscopy image. You can build a model of that. You can also do this in C. elegans for tissues. And in Arabidopsis, you can build models of tissue complexity there. So they're looking at different ways you can analyze networks in these different model organisms, where you can take data from some source and build a model out of it. So what they're looking at particularly is structural networks. So structural networks describe the physical associations between cells. This is akin to a roadmap which captures all the possible routes cars can travel upon. In tissue terms, this describes the potential communication between cells. Functional networks, by contrast, describe the observed communication and information flow between cells and tissues, akin to where traffic is observed in a road network. So they actually make a distinction between structural and functional networks. So structural networks are where we describe the physical associations between cells. So if we look at this figure, we see, the structural associations between neurons are the branching structures here and how they connect to other cells. So cell bodies are connected to other cell bodies through these projections, and we can map out those relationships in a structural network. A functional network, by contrast, is where you look at the flows between these nodes or these cell bodies, and so you want to ask the question, we know that these things are connected. What subset of these things are actually transferring information or transferring chemicals or transferring something from one node to another? We can think of this as the difference between, say, like a static network and a dynamic network, or a sort of a description of the structure versus something like the description of a flow. And so both of these types of networks are important for understanding tissues, and integrating them, of course, is important as well. Functional networks are constrained by physical networks. So the physical networks provide sort of the full architecture of the functional network. The functional network is usually a subset of the physical network. And functional networks are also dynamic in the sense that intracellular communication can be modulated across development. So these flows or these functions can be modulated in development from, as things are becoming connected, they kind of follow that connection, but they're not, not everything is active. Sometimes in development, function can reinforce structure. So in neurons, we have this idea that neurons get connected because they get connected with, say, like synapses and, axonal connections because of previous chemical cues that exist in the environment. So the nodes basically send out this chemical signal, and then those chemical signals are picked up by other neuronal bodies. They send out projections. They connect to one another because of some chemical affinity, and that sets up this structural network that then further sets up function at the synaptic level, so we can have a more refined chemical communication between the cells. That's basically the idea. And so all of that is hard to represent with just a simple set of nodes and edges. You need to have a little bit more in that network. But it's basically the same. You have to distinguish between the structural and functional networks. Okay. So then they talk about biological relevant network measures. And this is kind of interesting because a lot of the complex network measures that have been proposed have not been proposed specifically for biology. So, a lot of times, like things like between the centrality, clustering coefficient, shortest path, and degree are all artifacts of like what we'll call universal networks. That is, universal networks are networks that can describe biology, but they can also describe social systems, they can also describe technological systems, they can also describe just about anything else that forms a network, any set of interactions between things. And so we can have all sorts of networks that form topologies. Those topologies can be the same, they can be described with these statistics, but they're not biologically specific. They have this universality about them. That is, that you can use these measures and these topologies, you can describe them across a wide range of systems. Now, the problem with this, of course, is that in biology we have specific things happening that we would like to understand that maybe aren't well captured by these basic measures. So, like shortest path across the network is basically where you go from one edge of the network to the other edge of the network, and you calculate out sort of the shortest route or the most efficient path through that network. So that's what we mean by shortest path. Now, is that a description that is, that's often a description of any sort of optimizing process or optimal process. So, for example, we're interested in the shortest path not because it necessarily describes anything interesting, but because it gives us this criterion for optimality. If we have a flow of a chemical or a flow of information, what is the shortest path? How can it happen in the fastest amount of time? And then we can indeed find that path and predict some sort of flow. The problem is in biology, we don't often need to know the shortest path. We may need to know the shortest path if we want to know something, what the optimal paths to the network are. But sometimes we want to know other kinds of paths, longer paths, circuitous paths, paths that do some specific thing. Sometimes these things have evolved in ways that aren't optimal, and they're suboptimal. And so we want to understand those as well. Now, some measures like degree are interesting and relevant because we need to know, say, like the general degree of the network. So it's, this is where you have a node and you want to know how many other things it's connected to. So that's very useful in a lot of biological networks and understanding that degree for each node, but also the scale-free behavior of the network, which is where you have many different degrees across the network and classes of degree for specific nodes. And what those look like, what those identities of those nodes are, and so forth. The clustering coefficient, of course, describes where you get the most connections. And so in different parts of the network, you have high connectivity. In other parts of the network, you have low connectivity. And the places where you have high connectivity are what we call clustered, and we can describe it with a coefficient. That's very interesting from the standpoint of sort of aggregation. And of course, in biology, we're always interested in aggregation, especially in tissues. We're interested in sort of where the action is, where the communication is happening, that sort of thing. So that's a good measure. And then between the centrality is where you have this sort of, how many paths run through a certain node. So it's like how central is a node in terms of different parts of the network. So in this case, you have these nodes on the edges of the network, which have low between the centrality because they're not connecting anything. So they're just kind of the terminal nodes in this network. You have these other nodes that are medium, which means that they connect, say, one or two nodes in one direction and the rest of the network in the other direction. So they're kind of peripheral, and they have a medium between the centrality, and then nodes at the middle of the network that connect a lot of things to a lot of other things have a high between the centrality. So that's actually very interesting in biology, especially in tissues, because you can see sort of the center of mass of that network. And in like if we're interested in something like tensegrity, it might be a good way to look at like where the critical nodes are for maintaining ultra stability or stability. If we take those nodes out, do we remove the stability of the network? of the structural network. So these are all interesting measures, but in previous work that we've done in the group, we've proposed that there are other measures. I'll have to pull up the list of proposed measures. I can't remember if we have this in a working paper or if we have this in one of the papers we published. But we've talked about this in the past. And so there are these universal sort of measures that are interesting. Some are less relevant than others to specifically to biological networks, but some, especially in development, but some are, there are other measures that we can use in this case. So, like I said before, we have this idea of efficiency of transport that we want to capture in tissues. That's important. In multicellular networks, we might look at local and global efficiency measures. So we have these local and global measures. We're interested oftentimes not only in the entire network, but also in small compartments of the network. And so, we might be interested in this entire network, or we might be interested in this part of the network, this local part of the network off to the side. And the reason we want to do that is because, if we think about a network as sort of like, it could be hierarchical or it could be like regional, where things happen in one part of the tissue, move to the center of the tissue, and then, propagate throughout the rest of the tissue. That's all sort of a biological control that you can't easily describe by just describing the global state. So if you describe what's happening throughout the network all in one unit, like if we said we just want a network-wide measure of something, that only describes what's going on globally. There are a lot of things that happen locally in different parts of the tissue that can add up or accumulate over time and result in some global phenomena, like that the tissue like breaks apart or it suffers a massive injury or something like that. And we can't describe that unless we've described these local processes that have gone on before that sort of accumulate and give us that result. So this local versus global aspect of networks is very important. So we have these examples from Drosophila, from human tissues, from Arabidopsis, and we have these different types of things from bone, from the embryo in Arabidopsis, and from what's going on in Drosophila. So in Drosophila epithelia, we have this topology where we can examine this using network-based approaches. We can use the same type of model we've used in plant epidermis. In Drosophila epithelia, you have very specific structures. So we have to establish 6 neighbors. We have these cell division planes that, we have this growing network, so cells divide and then form nodes. Those nodes then get connected. And this way, local division rules lead to complex emergent tissue properties. To decipher morphogenetic mechanisms, it is, however, interesting to investigate differences between epithelia for different regions or developmental stages of Drosophila, and to compare those with epithelia from other species. So we, in doing things with development especially, we need to have networks that are growing networks. This is something we worked on in Google Summer of Code last year. We need to have growing networks, but we need to have network models that go beyond just like nodes and edges. So we need to have things like, gradients and growth processes and all these things kind of put together. It's not enough just to have nodes and edges. We can have hypernodes, of course, as we also did in Google Summer of Code last year. And so all these things need to be put together in sort of this meta model of developmental networks. They talk about Drosophila epithelium. They talk about the connections with some of the things that they're doing in Arabidopsis. Talking about cell differentiation patterns in the mouse embryo and in cancer tissues. And so you get these kinds of systems where they're heterogeneous. They have wide-ranging effects. Sometimes the local structures lead to changes in the global structures. there's a spatial distribution of things in tissues that are interesting to consider. So, not every cell is simultaneously doing something. If you think about the SIR model in epidemiology, you get nodes that are infected, you get nodes that are recovered, and there's a distribution of those at each time point. So it's a dynamic graph that changes over time, and there's some process that spreads throughout the network. And so, with cancer, you have a similar situation where cells can become cancerous, or cancer can spread throughout a tissue. You would model that using a sort of SIR model where a cell becomes sort of cancerous, or the cancer at least spreads that spatial compartment. And then you look at how many spatial compartments, or the spatial extent of that cancer conversion happens. And so you can then calculate network statistics, you can calculate local versus global dynamics, and so forth. They talk about cell division rules here, which are important for kind of for understanding cell division, but you have to then use a division rule based on that to sort of understand the topological state of a network. So again, this is something that we've kind of considered in our group, and this is where you have different ways that a cell divides, different things that go into a cell dividing, and you can incorporate that into the network. So they actually do this comparison in bone, where they look at the topological comparison of networks for lamellar bone versus woven bone. And so you can see that in lamellar bone, it's less densely connected, but with more optimized mechanisms for efficient transport. So in woven bone, you don't have these efficient transport mechanisms, and you also get this high betweenness centrality, which is aligned along paths that connect different lamellae and lamellar. So lamellar bone is different in a number of ways. You can describe those ways in this sort of network analysis. And so we have these different developmental rules. We have the first one is where we have mechanical forces acting upon a tissue. We have resulting constraint of space, but at the same time we have cell division. So we have apoptosis for cells that get kind of squeezed too much, and they disappear from the network, and the network then has to reconfigure. The second rule is the sort of movement leading to shape changes in the network. So as cells migrate collectively, they kind of stretch out, or they move away from one another. It stretches the network in a different way, results in shape changes in the network and topological changes. You also get this, in C, it's an example of neuronal osteocyte topology. So these are cells that do various things. They can bifurcate and split, they can merge, they can terminate, or they can branch. And so this is describing the growth cones of osteocytes or neuronal cells, where they can do these different things in their formation, and they affect how cells will be connected later. And so finally, getting to the generative models for tissue network development, there are a number of different underlying models. As we know, we have these different demands of development. We have growth, we have sort of, removal and addition of nodes. We have this, extra network topology mechanism for sort of bootstrapping developmental change. So all these things that we need to incorporate. Now they propose a number of different types of models here. The first one are Markov models. So, describing these processes as discrete Markov chain models. It describes the evolution of a system as a series of states in which the probability of transitioning to the next state only depends on the current state. So you have this sort of model of change that depends on the current state, and it produces the next state, and so forth. In the case of tissue networks, the state of a tissue has been described as a vector of frequencies of no degrees. So this is where they talk about tissue networks and how they describe this state. Hence, an entry in the vector contains the frequency of cells with a specific number of neighbors. Proliferation rules specify the probability of an I-sided mother cell to produce a J-sided daughter cell, and thereby determine the probability for the system of transitioning from one state to the next. So this is where, we have these rules for producing, we have sort of the current state depending on the previous state. We could map that to mother cells producing a daughter cell. So the state of the daughter cell is dependent on the mother cell. It's not dependent on the mother of the mother of the mother cell. And this limited amount of memory, sometimes is quite useful. While there are there's epigenetic memory, while you do have these different aspects of, um, you know, you have to take into account contingencies in development. You also have these sort of immediate things that contribute to network dynamics. So, using a Markov model allows us to remove a lot of extraneous stuff and just focus on the most important relationships. We also have agent-based models. That's a second model. An agent-based model is, a grid where it represents a complex system, a complex set of global behaviors from the interactions of local individual constituent cells. So these are a set of autonomous agents you can program with rules, those rules that are executed at the local level, interactions between cells, that then kind of allow you to emerge global states. So when you look at an agent-based model, you have each cell operating individually, interacting with its neighbors, and then producing this global state that you can observe in the whole of the model, in the entirety of the grid of the model. So they talk about rules in agent-based models, which can be discrete, such as if-else statements, or continuous, which are based on differential equations. So we can use our differential equations, we can use our if-else statements. We can derive those from the dynamics of cells. We can derive those from the dynamics of the internal workings of the cell, like the genome, other kinds of interactions. And we can do this for all cells, and they provide an input, a set of inputs and outputs for each cell, and they interact in that way. So, we can provide various amounts of spatial detail in an agent-based model, and we can take what's done in agent-based models and transfer it to complex networks. Okay, so apart from the basic tissue topology, the distribution of cell fates is also of major interest in developmental systems. In a centroid model, cell fate markers can be added as properties to each cell. In this case, cell division rules determine how a fate is passed from the mother cell to the daughter cell. Testing different inheritance rules in a centroid model, and subsequent comparison with experimental data, reveals that symmetrically passing on the same fate from mother to daughter best reproduces the cell fate patterns during primitive endoderm differentiation in ICM organoids. So this is something that, this is inner cell mass organoids. So this is where they kind of determine some of these rules for the agent-based model based on the biology, based on how they replicate these sort of global states in different biological systems. So they have these two different versions of agent-based model, the vertex model and the centroid model. So basically you have, you can have the cell as a vertex, you can have the cell as sort of the centroid, and so let's say in the vertex model, each cell is represented as a polygon. This corresponds to a tissue network in which the nodes describe the polygon corners and edges of the polygon sides. So this is where we're describing kind of more of the shape of the network, and that's the network instead of interactions between cells. Forces acting on a cell encompass cell elasticity and adhesion, and cellular packing is determined by stationary and stable network configurations that fulfill a force balance. This is the sort of thing we're trying to do with our biotensegrity models. So there are a lot of things you can use a vertex model on, a lot of types of problems such as packing geometry, and then, in some cases like the Drosophila wing disk, we can look at node degree and some of these processes where the imaginal disk is producing these highly patterned sets of cells that interact in this tissue structure. So this is sideways, but this is table 1, and it basically talks about the different types of models, the biological question that you can answer with it, the mechanism it's sort of approximating, and the modeling approach you might use for this. So there are a lot of different types of agent-based model, Markov model, and other types of centroid models, agent-based models, and of course other types of uh, uh, differential equation models, such as hybrid grid continuous three-dimensional model, continuous two-dimensional, three-dimensional model, and a mean field two-dimensional model. Okay, so that's all for that paper. And so I think we've covered 2 papers today that have kind of gone over maybe some of the generative aspects of development, but also how we can plug development into more sophisticated models of C. elegans and how we can produce these sort of Turing complete models of simulating C. elegans. So that's all for today. Thank you for paying attention, and I hope you learned something.